Okay, this is for biology, and I wanted to go over the last thing that is in the handout on evidence for evolution. Looked at fossil evidence for evolution. We did that yesterday. We also looked at the anatomical evidence for evolution. We looked at homologous structures and vestigial organs, and looked at some examples of all of those things. The hottest area, the most significant area right now in all of biology, really, but evolutionary biology does apply to that as well, is looking at molecular biology and how it's impacting our understanding of the relationships between species. So, last handout has a guide for molecular biology, and we're going to be looking at a protein called cytochrome C. And cytochrome C, you could read this in here, but I'll explain it as well, as it is a protein found in the mitochondria of most animals. And we're going to look at the sequence of the amino acids in this protein and compare it across different species. So it's got I've got a handout on the back here, and I can show you what that looks like. There's 14 different animals that are listed on here, as well as other organisms. So we're going to be looking at just a few of them. We're going to look at humans, going back to this side, dogs, kangaroos, rhesus monkeys, tuna, bullfrogs, snapping turtles. The dog's already been done for you, so you've got some data for that. And we're going to be looking for differences in the amino acid sequence between humans and each one of these animals. So the first thing you're going to do on step one here, you're going to find the humans, the rhesus monkeys, the kangaroos, the snapping turtles, bullfrogs, tuna, on the amino acid sequence on the back of this page that I just showed you. And you're going to underline those names. So those are the ones you're going to be looking for. And then you're going to go through for each one. And this takes a little bit of time and some patience. And using a highlighter, and I'll have some highlighters handy, mark any amino acids that are different than the human sequence. And when you're finished, you're going to record that number in this data table here. The dogs have already been done for you, and I'll demonstrate that on the dogs. And then we've got this table down here. This is a cladogram. Cladogram is the way an evolutionary biologist shows the relationships between organisms. So humans are here, and next we have rhesus monkeys and kangaroos and snapping turtles and bullfrogs, tuna, and lamprey. So what this is showing is how closely related these organisms are. So humans are more closely related to rhesus monkeys, and we diverge from them and have a common ancestor with them more recently than we did for kangaroos, which is a little bit further back. So further back, we have a common ancestor for kangaroos and humans. And if you go further back, there would be a common ancestor for snapping turtles and humans, and so on and so forth, all the way back to the lamprey. So we're going to be keeping track of these sequences and then comparing them to this cladogram. It'll be on the last page, and looking to see if we can see some patterns in those differences. So let's look at how I want you to collect this data. So going back to this chart here, and hopefully you can hear me okay. So we've got humans up here, and humans and chimpanzees really have no significant differences. We've got the rhesus monkeys here. You can find the others, but here's the dog right there, okay? So we're going to be looking for differences between the dog and the human. The first thing you'll notice is some of these have some starting amino acids at the beginning and some don't. Those would count as differences, by the way. So in the case of the human and the dog, they both have the same number of blanks there. So I don't see any differences there. You'll see the same thing at the end in some cases. Some have an extra base down here at the end. Some don't. Here's a key telling you which amino acids are represented by which letter. So we have A for alanine, C for cysteine, and so on and so forth. So going through the dog and the human, I can find, go through and look and see that's the same, that's the same, that's the same, that's the same. So you just kind of go along and look for any amino acid that's different. Sometimes helps to put a paper underneath these to see, and it looks like here's one. So that is one difference right there between the human and the dog. So there's one amino acid sequence. So, and if I keep going, and actually, I don't think you need to do it on the human. I think you just need to do it on the dog. The human will get kind of messy if you keep doing that. So just highlight the difference on the dog, and then keep going. And if we count, and assuming this is correct over here, and assume it is, and you don't have to double check the dog, but make sure you check against one another for the others, there should be 10 amino acid differences in the dog, okay? When you're done with that, you're going to fill out this page here. So here's the cladogram again. And in these hexagons here, you're going to put the difference, the number of amino acids difference in that you collected on the previous data. So just copy this over from the other data table. So how many amino acids? And for the dog, I think we already have one. Dog's not on here, but it would be 10 if you had the dog. And we would fill in 10. And then there are a few questions you can answer, assuming we don't get to those. Remember that on Friday, we are going to start looking at your presentations. I've been looking at that, and I know who's going to be doing which topic. We'll talk about the presentations. Those will be due. You'll have a week to work those out. This will be due for Friday of the following week, and hopefully we'll have time to get through all of those. So if you have questions, you can kind of leave an email for me, and I'll try to keep checking that and try to get back to you during class time. I'll also call in and check in and see if there are any questions that way. So good luck on that.